And also, since we're already running behind, we'll go ahead and get started with our first speaker, Dr. Joe Roche, who is a professor in veterinary integrated bioscience. And he's going to be talking about corpus luteum survival and establishment of pregnancy in ruminants and molecular and cellular revisitation of concepts. All right.
Again, good morning to everybody. I thank it's off. I thank the organizers, mainly Greg Johnson, giving the opportunity to come and present our recent data sets. Corpus luteum survival and establishment of pregnancy in ruminants, a molecular cellular revisitation of concepts. I dedicate this talk to my beloved vendor, colleague, and friend, Dr. John McCracken. A few slides about him. Dr. McCracken is a DVM, veterinary surgeon, and PhD in endocrinology, 1935 to 2019. Dr. McCracken wisdom in corpus luteum physiology and endocrinology and ovarian uterine transplant is legendary. So UConn released about his uterine transplant, how that was used by the Swedish scientist to make the uterine transplant in human. Also, Dr. Fuller Baser wrote about him in Biology of Reproduction. You can read that. It is 1972. He gave that picture to me, and 2008 and 2018. So, prostaglandin research. This captured 2 Nobel Prizes, 1970 and 1982. This is the person Dr. John McCracken collaborated in 70s, Samuelson. He is in 72. John McCracken discovered prostaglandin F2 alpha as a luteolytic hormone in ruminants in 1972. This is a major breakthrough during that time. He published his research in Nature New Biology by that time. Today we call them Nature. So he collaborated with this person in Sweden to do the mass spec, and by doing the mass spec in 1970s, he identified PGF2 alpha as a luteolytic hormone. So he's known for his 2 models. One is ovarian transplant in the neck. Actually, he removed the ovaries from the abdomen. He put in the neck anastomosis with the jugular vein and the ovarian artery. And he used this model for 40 years in his field. No one did this transplant model until today, except him. He taught those techniques to me last 10 years. So I learned some of them. Also, he developed a 5-pulse model. 5 pulses are required to induce luteolysis in ruminants. So this is the one standing in the field forever, almost 40, 50 years after he discovered that PGF2 alpha is luteolytic hormone. He wrote a review in Physiological Review 1999. This has 750 references in that. So it has all the mechanism about luteolysis in ruminants and across all the species. If you need to know about that, his work, This will be the better paper to read. I met Dr. John McCracken in 2002 when I was a graduate student in one of the SSR conference. So I invited him to Texas. This is my home in 2006. He came to me and explained how we can do this uterine transplant model and all the catheterization. It's abstract. So he taught me this surgical technique. So he showed me how we collect the uterine ovarian, how we identify uterine ovarian plexus, how we can catheterize the uterine vein, how we collect blood from the ovarian artery, and a lot more. So Dr. Makarak and I worked together for last 15 years. So we were able to secure 3 fundings continuously from 2018 to 2023. So last email he sent it to me because I visited him after SSR 2017. He asked me to do a reevaluation of estrogen receptor and oxytocin receptor in the sheep. You might want to look at figure 10 in McCracken et al. 1999 physiological reviews because we had some discussion on that. He wrote me, you need to revisit that. The last mail I received from him after a year, 7/24/2018, after 3 weeks of hospital for a recurrence of my bronchial pneumonia, I have moved to assisted living in Braywood. I am recovering slowly, but I am hanging in there. Here is the title for our next paper. He said, upregulation of the endometrial progesterone receptor by the trophoblast established pregnancy in sheep. This was his last thought about this progesterone receptor story. And I'm still working on that, how we can finalize that. So Dr. McCracken wisdom in corpus luteum hegemony, Dr. McCracken contribution to corpus luteum physiology and endocrinology stand forever in the scientific community. I will greatly miss him. He gave me this quote, I modified to the, he gave this quote, one of his mentors gave it to him. I took that quote and modified and fit into him. What the initiator know and his patient integrity and devotion to exact observation in 1970s, they have made this step that is my current research possible in 2018. Scientific background and scientific premise. In ruminants, endometrial PGF12 was secreted in a pulsatile pattern at the time of luteolysis. This is the luteolytic hormone, causes functional and structural luteolysis. Functional luteolysis denotes decline in progesterone, and structural luteolysis denotes physical involution and regression of the luteal tissues. The relationship between functional and structural luteolysis has not been clearly identified, but we believe that functional luteolysis appears to precede the structural luteolysis. Luteolysis means regression of corpus luteum, so that progesterone will go down, so that animal comes to the estrous cycle. When the corpus luteum persists or maintained, progesterone will be maintained, animal will maintain the pregnancy. During the estrous cycle in sheep and ruminants, endometrial PGF12 are released in a pulsatile pattern. At the time of establishment of pregnancy, interferon secreted by the conceptor suppresses these pulses. He established that 5 endometrial PGF12 pulses of 1 hour duration over a period of 48 hours at 8 hours interval are required to cause complete CL regression consistently in sheep. This is the dogma here, because 5 pulses are required. That should be 8 hours interval for 48 hours. If it is less, CL is not regress. If it is more, CL is not going to be regress. This is one of the major discovery in his research. So in our laboratory for the past 10 to 12 years, we are working on this corpus luteum physiology and endocrinology. Endometrial PGF12 induced by pregnancy is transported locally to the ovary, which increases luteal PGE to biosynthesis and activates luteal EP to EP4 signaling, thus protecting the CL during establishment of pregnancy. So in this paper, we reported that luteal EP to EP4 signaling is very important for the maintenance of the corpus luteum. Next paper followed by, in this paper we have identified what are the proteins are regulated at the time of natural luteal lysis in sheep. So we identified a lot of survival pathways are going down and the apoptotic pathways are going up at the time of natural luteal lysis in sheep or in cow. The question based on our previous findings, the question we asked, how the CL is maintained and survived during establishment of pregnancy in sheep. During natural luteal lysis, the CL is regressed spontaneously in response to PGF-12 pulses. These pulses are transported from uterovarian vein to ovarian artery. In hysterectomy model, sorry. In hysterectomy model, there is no PGF-12 pulses, so the CL is maintained. In ovarian transplant model, there is PGF-12 pulses, but absence of local transport because ovary is in the neck, the CL is maintained. At the time of establishment of pregnancy, absence of endometrial PGF-12 pulses because of the concepted secretory interferon, the CL is maintained. During natural luteal lysis, if we give exogenous injection of PGF-alpha, the CL is regressed. In hysterectomy model, if we give exogenous PGF-alpha, the CL is regressed. In ovarian transplant model, if we give exogenous PGF-alpha, the CL is regressed. In pregnancy, if you give exogenous PGF-12 during day 13 to 16 of the uterus cycle, the CL is resistant or maintained. So what's the story behind this? How the CL is resistant to this PGF-12? So based on that, we asked three questions here. How the CL maintained in survey during establishment of pregnancy in sheep? Hypothesis 1, restoration of endometrial PGF-alpha pulses may regress the CL of early pregnancy, because that was the discovery made. So we thought, Dr. McCracken and I thought, okay, let us restore the pulses. Let's examine what happened to the CL. Hypothesis 2, selective inhibition of luteal PGE to EP to EP4 signaling may regress the CL of early pregnancy. Hypothesis 3, combination of both. Maybe it's two mechanisms are required. Absence of pulses, but at the same time, regression of EP to EP4 signal. So, we did this experiment. Jagun is here. Jagun was working with me during the time. So, we both work hard on this experimental setup. Jagun knows all the drawback, all the hard times we had with Dr. McCracken and setting this experiment and all. So we had 4 groups. Group 1, in the jugular vein, we put the saline, and intra-ovary infusion, we put the vehicle. In group 2, in jugular vein, we put 5 pulses of PGF-alpha. In the ovary, we put vehicle. In group 3, in jugular vein, we gave 5 PGF-12 pulses. In the ovary, we gave EP to EP4 inhibitors. Group 4, we give 5 saline pulses in the jugular vein and EP to EP4 inhibitors into the ovary. So we designed 2 by 2 factorial experiment. So where we give, this is the saline or PGF-12 infusion. We started between day 14 and 16. The first pulse, we give day 14, 8 a.m. Then the 16 hours interval in between first and second pulse, we give 12:15, 12 a.m. in the midnight. Third pulse, day 15, 8 a.m. in the morning. Fourth pulse, day 15, 4 p.m. in the evening. And the fifth pulse, day 16, 12 a.m. And day 16, 4 p.m., we did the necropsy. The EP to EP4 inhibitors, we put on day 10 and maintain throughout the pregnancy. EP2 inhibitors, we right away give into the ovary. So if you look at this between day 14 and day 16.5, this is the way we give pulse 1, pulse 2, pulse 3, pulse 4, and pulse 5. This is just we adopted Dr. John McCracken model. So this is the way we put this ovarian infusion. We put the asthmatic mini pump in this broad ligament. We suture in such a way that the catheter doesn't come out. And we identify the spot is a avascular spot in the ovary with the catheter inside ½ inch and suture them back. So this will pump required concentration of EP2, EP4 inhibitors for 7 days. These are the cages Dr. McCracken designed after his visit to Australia in 1970s. Then after he retired from UConn, he donated to us. So we have all this in Astra. This is the way we did 5 sheep or 4 sheep at a time. This is the pump we have it, so we infuse with the jugular. So what we did after every infusion, we measured PGFM in the jugular vein that to confirm that we are giving PGF to alva, it is entered into the body and we would be able to detect the pulses. That's the first pulse. So 0 hour and the midpoint and at the end. So we took 3 points here before, 30 minutes and 60 minutes. So we measure here first pulse, second pulse, third pulse, fourth pulse, and fifth pulse. We made sure that the pulses are in. If you look at that saline pulse, there is no pulses here. PGF to alva pulse, we can see a clear pattern of pulses here. PGF to alva pulse plus EP2, EP4I, we have the clear pattern of pulse here. And saline pulse plus EP to EP4, we don't see the pulse. So our model is working. We are able to detect the pulses. Then we measure the progesterone level. In saline, these are all pregnant sheep. In saline, the progesterone is maintained. PGF to alva pulse, the progesterone is maintained. That means restoration of PGF to alva pulses fail to induce luteolysis as we see at the time of natural luteolysis in cyclic sheep. And PGF to alva pulse plus EP to EP4 inhibitors, this decreases the progesterone level slowly. EP to EP4 inhibitor alone, they did not decrease the progesterone. So this is the way we measure between the pulses at 8 hours interval. So as Dr. McCracken observed several years ago, we observed the same thing. After the first pulse, progesterone goes down in all the group, here also. But after 16 hours, come back, the CL become refractory to this PGF2 alpha, then it's maintained in all these groups. But only in PGF2 alpha pulse plus EP4 inhibitors, this progesterone is going down. At the time of necropsy, we collected the CL and we weigh them. Also, we measure the luteal volume. These are all no change here in these 3 groups. Only the group received PGF to alpha pulse into the jugular vein, EP to EP4 inhibitors into this ovary. They regress the volume and weight. So this is correlated with what we see in progesterone profile. Then we took some picture of the corpus luteum because this is the morphology, saline pulse, PGF-12 pulse, PGF-12 plus EP2, EP4. This is the one going for regression, and EP2, EP4 pulse alone. So we flush the uterus. This is the technique we learned from Dr. Baser. So we flush the uterus with 20 ml of saline and make sure that the sheep is pregnant. Also make sure that they have a normal morphology. To me, all the conceptors is normal in morphology. That means they are healthy conceptors inside the uterus and the CL is regressing here. So we went to structural and functional luteolysis pathways here. First thing, we measured progesterone pathways. What happened to P450 star 3 beta HST? We see some effect on P450 SCC and star by this group, but not with the other treatment on this protein expression. Also, we measured this where these proteins are localized. Most of the time, we see that these proteins are localized in large luteal cells as well as in the small luteal. This is a star. Then we look for luteal PG biosynthesis and signaling. This is the place we begin to see the difference, because at the time of natural luteolysis, all these survival proteins will go down. However, in the CL of early pregnancy, resistant to this PGF2 alpha, most of the protein what we see here, FP, EP1, EP2, EP4, PGDH, all these enzymes are stable. When you do this combination of two, some proteins are going down, but we can see that in the other treatment too. EP4 is example, both are reducing. FP only this treatment. If you go above that, AKR1B1, the PGFS synthase is going down in both. So we are not able to conclude what's going on here, because this is the only one causes the regression and progesterone goes down. But we see the proteomic changes in the 2 treatment or 3 treatment together, or at least 2 treatment in combined way. Then we look into the luteal cell survival and apoptosis pathway. If you look at the ERK1 and 2, it's going down in both treatment, but progesterone go down only in this. Then AKT is going down here only by this group. It looks like AKT is the one is supporting or maintaining the survival of the CL. When to the beta catenin, NF-kappa B is all absent in this all 3 pathways. The treatment here also is going down. Here, this result also, we are not able to conclude which protein is maintaining the survival. Then we went for all these BCL to caspase 3 apoptotic things. Again, we see the same trend. If you look at the PCL2, it's decrease in all 3 treatments. BCL-XL, 3 treatment. BAX going up in 3, BAD going up in 3. XAAP, this, whatever we see the protein changes, we pick up this protein because of these proteins are involved in natural luteolysis during the cycle. We reported that these proteins are important. But when we do the pregnancy, we are not able to see the same result. It tells us that something going on with the CL, the mechanism is not known, which is supporting the survival of the CL. So after this, we move into this RNA-seq approach to do this discovery-based research. All time we say fishing expedition, now NIH says discovery-based research doing omics technique. So we did the global transcriptome expression using RNA-seq approach. We were able to detect about 22,000 transcripts, and we divided them high expression, moderate expression, and low expression. Several genes fall under this group. Heat map showed here, this is the pregnancy, and this group, 3 of them have changes compared to the control group. Some changes are here, some here, some here. And this change is little bit unique to this group because they are difference in the expressions of cluster of genes in this place. So it gives some idea that there are some specific gene expression in this group compared to the other group that we do not know yet. So we moved further into that. So 16 pregnancy with PGF2 alpha, totally 245 mRNAs are regulated, transcripts are total mRNA regulated. Among them, 72 are upregulated and 173 are downregulated. In the combination of two treatments, 155 mRNA is regulated, 64 are upregulated, and 91 are downregulated. In EPI group alone, again we see 139 mRNA upregulated, among them 40 upregulated and 99 downregulated. Then we did this Venn diagram here, cutoff fold is twofold. When we see that total of 72, there are 17 commonly regulated here, and 10 is common in all three treatments, and 9 between PGF12 and EPI, and 4 between here to here. In most of the time when we do this kind of research, we will be focusing on what is the gene commonly regulated among this group. But in this treatment plan, in the question we are asking, we focused on what happened, what are these 33 genes just regulated by this combination of these two treatments. So these are the 32 genes we have. Whatever you see ENSO, they are new genes because those genes are not characterized in sheep or in cow so far. So these genes, so I pick up one of the genes that's a highly expressed gene to look into that what's going on. PubMed doesn't give any result to us because these genes are not characterized in sheep. Based on the human counterpart, we come up with a protein called calthrine. Calthrine is a heavy chain linked domain containing protein because all these genes are protein coding genes. So if you look into that, these have a several network connection here. And when we put this gene into IPA, just to predict what are the signaling or crosstalk it has, they have so many crosstalk in this. So we are going one by one to pick up what could be the gene or what could be the set of genes it could regulate or it could play a role in this main survival and maintenance of the corpus luteum during pregnancy. The same way we went for the down-regulated mRNA. So several genes are regulated, that are common, but we are going behind this 38 genes that is only regulated by this PGF-alpha EPI gene. Again, I pick up the other one is a 5.3-fold here. When I put in PubMed or Google Scholar, nothing show up in the sheep because it was not characterized or not known yet. So I pick up the second one, the FGG, and I put in IPA to see that what is going on. This is a globulin domain containing protein, both are them cytosolic protein, and it has so many interactions here. So how we can track them? Is there any way, because instead of going one by one, how we can track them? Then I asked them bioinformatics to agent genes module. So when we do them, there are 4 modules. This is the agent genes model. This is the correlation. So this is the MS plot. It shows that these genes are distinctly located. There's a distance between these, a green model, brown model, black model, and turquoise. This is the original picture the software give it to us. But if you look at that, I am a little bit confused about that, so I couldn't interpret that in the way I wanted to, how we can explain that easy to people. Then we made this kind of diagram to make it simple and easily readable. So black model gene, it is here. All treatment is going down. PGF-12 bring them down, combination bring it down, EPA alone bring it down. When we go for brown model gene, PGF-12 increases and PGF-12 increases and combination also increases and EPA also increases. When go for turquoise here, this PGF-12 bring it down, combination also bring it down at the different level, EPA increase. When we look for green model, PGF-12 bring it down, this combination is increases and EPA alone is bring it down. So when we look into these modules, what you have saw before, these are upregulated genes, only brown and green models are involved in the upregulated genes. So brown model 22 genes and the green model are 11 genes. When you look into the downregulated genes, just opposite, only black and turquoise model genes are involved in this downregulation. So if I put together here, so what this story tells us, not a single gene is responsible for this resistance of corpus luteum during the establishment of pregnancy. So there's a set of genes need to be upregulated and the set of genes need to be downregulated. So we are working on this to pick up the set of genes, what we can do, functionally silence them or overexpress them and to see what happened to this here. That might be our next grant we are getting. So in summary, restoration of endometrial PGF-12 pulses or inhibition of luteal PGE2, EP2, EP4 signaling failed to induce luteal lysis during early pregnancy. In contrast, combination of both treatments successfully induced functional and structural luteal lysis during establishment of pregnancy in sheep. Rescue of the CL during establishment of pregnancy in sheep is not only due to suppression of endometrial PGF-alpha, as we previously believed, but it is also due to increased intraluteal PGE2, EP2, EP4 signal. Functional luteal lysis is due to impaired progesterone biosynthesis, and structural luteal lysis is due to combination of multiple cell signaling pathways. So luteal rescue mechanism, that's the one interferon to or conceptor secreted interferon to suppress the endometrial PGF-12 pulses. This is luteal rescue mechanism. After the CL is rescued, I think the animal need to protect the CL by activating some mechanism in the corpus luteum itself. So that's the one we see here. There's a regulation of genes and proteins in the corpus luteum by the conceptors. These two mechanisms are required for successful establishment of pregnancy. Either one of the mechanism is not sufficient. How the conceptors regulates expression of these genes and proteins in the corpus luteum in ruminants, that will be the future research. So I thank USDA for continuously supporting our research from 2008 to 2023. That funding provided opportunity to support several trainees in the laboratory in the past. One of the important trainees from my laboratory is Dr. Jahun Lee. He worked with me from 2005 to 2016. So most of the C project, Jahun and I did together. And also I thank Dr. John McCracken for his contribution to this piece of work. Thank you. I'm glad to take if you have any questions.
There are people in our laboratories that are interested in veno-arterial signaling, and so the prostaglandin pulses get from the endometrium and somehow make their way into the ovary, right?
Yes.
How does that happen?
Okay, if I understand your question correctly, how the PGF-12 is transported from the endometrium to the ovaries.
Yeah.
So in sheep, is the endometrial PGF-12 transported to the ovaries to the local mechanism. There is anastomosis between the ovarian uterine vein and the ovarian artery. That's going together. But this is not the mechanism in human. human is completely different. This mechanism only in the ruminants. That's a local transport between the uterine vein and the ovarian heart. That's a coil together. That's an exchange between those. Some part of the transport is mediated by prostaglandin transporter protein. Some of them mediated by as a countercurrent mechanism.
Very interesting data. And I was going to ask if you're familiar with the model that Dr. Milo Wilbank has for cows, proposing that also there's some changes in blood flow during pregnancy that would actually protect the CL, would alter the transport of prostaglandin F2 alpha to the CL and actually have some protective effects on maintaining the CL due to major changes in blood flow during the beginning of pregnancy. Have you seen any of that in the sheep, and do you think that plays a role in the sheep?
Yes, we reported. Milo was very much interested when we discovered prostaglandin transporter in 2004. Then afterwards, we discussed, and he was very much interested to understand that further. So we reported that in sheep. I don't know, I forgot the year. Sometime in 2012, or before that, we reported that the prostaglandin transporter protein has affinity, selective affinity to transport either PGF2 alpha or PGE. That protein is expressed in the in the junction, UOV and ovarian artery junction, that protein is expressed. So we put the DITs into the ovarian vein, the plexus, we blocked the PGT protein for 4 hours. It affected this luteal maintenance or luteal lysis. So that protein expressed in this UOP ovarian plexus plays a role. Both laboratories supported the same findings.
All right, let's thank Joe one more time. Okay, now we'll move into the trainee presentations. Our first one is by Udi Betty out of a PhD student in Michael Golding's lab. He's going to be talking about prenatal alcohol consumption alters the sperm epigenome and leads to fetal growth restrictions.
I found it, yeah. Thanks. Good morning, everyone. I'm happy to be here to talk about some of my research here at Dr. Golding's lab. And my title is going to be Paternal Alcohol Consumption Alters the Sperm Epigenome and Leads to Fetal Growth Restriction. So I'll present some data on that. The first thing I'd like to mention is that there is now growing evidence that paternal lifestyle exposures and paternal environmental exposures such as pesticides, toxins, smoking, there's a lot of evidence now that these can lead to disease phenotypes in the offspring and lead to adult onset diseases in offspring produced by these fathers. So alcohol is a well-known toxin, and it's really well known that alcohol has effects on the next generation. And just for some statistics, in the US alone, 70% men drink alcohol and 40% of these drink heavily, that is binge drinking. And while few studies have focused on the singular role of alcohol consumption, there was recently this systematic review published 2 weeks ago that actually shows how paternal alcohol consumption has a higher links with congenital heart defects. And the odds ratio here is 1.4 out of 24 studies. So this is really fascinating. Now going back to what we were focusing on, fetal growth, we started work with this model where we had C57 black 6 mice, and we were giving them alcohol for 4 hours every day. And this is a well-established protocol called drinking in the dark, where if you give them access to alcohol as soon as the dark cycle hits, they really go for it and binge on it. And it really works. So we did this for 70 days, and the reason we chose that was because that encompasses about 2 spermatogenic waves in the mouse. And after that, we obviously mated them to female C57 black 6 mice, and then terminated pregnancies at GD 14.5 and in later study 16.5. So the reason we chose 14.5 was that it's easy to separate the placenta from the maternal interface at that stage, and then you can really characterize things like placental weight and placental efficiency. So in the first round, we did dissections at GD 14.5. Here what we found was that the gestational sac weight in both males and females was reduced. Also, the other parameters of growth, such as fetal crown rump length and fetal weight, were also reduced. Albeit in the fetal weight, it was more obvious in females rather than males. So we wanted to go a bit further and see if this growth restriction actually develops even more. So we went to GD 16.5 in our next study, and there it was really striking. So we had a 15% reduction in fetal weight in both male and female offspring. Also, crown rump lengths were reduced significantly, and we also had the overall gestational sac weight reduced in the male and female offspring. It was interesting to note here that the placentas of the males were slightly larger, and this led us to a little graph here called placental efficiency, and that's basically the unit of fetus produced per unit placenta. So you just divide the fetal weight by the placenta weight, and it was cool to see that placental efficiency went down in both male and female offspring, but especially in males. So knowing this and going forward, we wanted to see what was changing on the male side of things, what was changing in the reproductive system of the males from the alcohol. And that's when we started looking at gross morphology, such as testes weights, epididymis weights, seminal vesicles. And then we also went into, we did some sectioning and looked at seminiferous tubules in the testes. And we found that there was no difference. This 10% alcohol dose did not make any difference in any of these parameters. And also plasma testosterone was equal in both control and alcohol sires. So this was really important to mention because there have been other studies. There have been studies using 25% alcohol. There have been studies where they do a garage method of alcohol for a shorter period of time that have reported differences in the testes weights, basically a reduction in testes weights, a reduction in testosterone, and even in the seminiferous tubules, there's 2 papers out there that show the seminiferous tubule size decrease, which correlates well with sperm production too. So this was important and a good finding for us because what we're showing here is that there's no difference in the male reproductive tract, but this low dose of alcohol is actually causing differences in the fetus downstream. So from here onwards, we wanted to look at the sperm morphology and the sperm parameters. So obviously, we looked at sperm count and motility and viability. So there was no difference there. Tunnel assay is just a double-strand break, DNA double-strand break assay. So we measured DNA damage with that, and clearly there was no difference in DNA damage. CMA3 is chromomycin A3 is basically competes with protamines for binding in the minor groove of sperm DNA. So as many of us know that sperm DNA is highly condensed and it gets packed with protamines rather than histones. So if there's a protamine deficiency, then you see CMA3 pop up. And as there were no differences there, so we can conclude that broad chromatin structure was maintained. So going forward, we say that there's no difference in male reproductive tract, there's no difference in the sperm morphology. So we wanted to look at what's changing that's not DNA related, but it controls gene regulation. So that's where we delved into the epigenetic studies. So the first finding in 2017 we had was when we were looking at sperm DNA methylation. So a lot of studies have reported changes in DNA methylation, albeit between change at one methylation site at 5% or something like that. So we wanted to look if there was any major changes in DNA methylation. That's why we insisted on looking at 25% changes, and we also increased the coverage. And at this, at these parameters, there was absolutely no change in DNA methylation. You can see here that global methylation was not different. So most mammalian methylation is at CpG, even though there are methylations at Cs away from Gs or are not located next to Gs as well. But CpG methylation dominates, and there was no change in that. And here we have the Pearson correlation chart for the 3 alcohol samples and 3 controls, and there was no difference. They all correlated very well with each other. And here's just another description of where CpG methylation is found in the mammalian genome, especially in sperm genome. So sperm DNA methylation was changed, but there's more to epigenetics than just DNA methylation. So we started looking at the sperm non-coding profile, and this is where we found some interesting data. So the gray here is tRNA fragments, and the orange is piRNAs. So in mature sperm, you have this profile where a large proportion of your non-coding RNAs are made up of tRNA fragments, and it's roughly 60%. And the piRNAs form about half of that population. So when we compared our controls to our alcohol sperms, what we found that there was a slight, ever slight shift in the tRNA fragments, a decreased abundance of tRNA fragments, and an increased abundance of the piRNAs. And when you look at the ratio, the change in tRNA fragments over piRNAs was decreased. So why I thought this was important is that it is now well known that the sperm non-coding RNA population actually changes as spermatogenesis occurs, but most of it, the most dramatic changes happen at the very end from testicular sperm going through epididymal maturation. And this is just, so the green here is the piRNAs, and the red color is for tRNA fragments. So the tRNA fragments really dominate towards the end of spermatogenesis, especially during the epididymal maturation phase. And seeing our data, looking at how tRNA fragments were slightly reduced, my hypothesis going forward would be that the alcohol led to some kind of a delay in epididymal maturation, or maybe altered how the tRNA fragment abundance was supposed to proceed. So this was really interesting to find. And the other part of the small non-coding RNA profile that we also looked at was microRNAs. So even though the microRNA sliver is really tiny here, we did find an increase in microRNAs. And for the data set that we had, this increase was about 30%. And here's another heat map showing in the various controls and the alcohols how that looked. So you have a lot of microRNAs going up, and a lot of them that were supposed to be higher that are reduced. Especially in our data set, we found 3 microRNAs stand out: microRNA 142, 21, and 38. So while 21 and 30 were increased, 142 was decreased. And here we come into that epididymal maturation story again. So this is from the same paper I showed previously. They also characterize microRNAs, and microRNAs actually change quite dramatically over the course of spermatogenesis, spermiogenesis, and then also epididymal maturation. And it's relatively the same shift, same time when testicular sperm are released, and that's when most of the shifts happen. So again, my hypothesis again going forward would be that epididymal maturation is where we need to be focusing our attention for the alcohol exposure. And interestingly, the 3 microRNAs that we did find altered were also in that data set that changed during the course of spermatogenesis. So we don't really know a lot about what happens epigenetically at epididymal maturation. What we do know is that sperm get the cytoplasmic droplet migrates, and sperm suddenly increase in their motility, and they're actually capable of fertilization. There's a debate on whether caput sperm versus cauda sperm is enough for fertilization, but there is mounting consensus that there's a class of RNAs that are produced in the epididymal epithelium that basically are secreted as vesicles or epididymosomes, and they get fused with sperm during the course of epididymal maturation, and that's what leads to that shift that we see. And this has been done in a few studies now, and it's also been seen that the epididymosome RNA population resembles the deficit that we see in sperm maturation. So going forward, so my conclusions are the same, that paternal preconception alcohol leads to fetal growth restriction, and somehow the epigenetics of the sperm are at play here. Going forward, we probably want to look at epididymal maturation, so we narrow the alcohol exposure window from 70 days to maybe a shorter time that represents the period of epididymal maturation, and see if those are where all the changes are happening, and also sequence the RNAs from epididymosomal vesicles to see if those are changing with the alcohol exposure. And another step of the epigenetic program that we're currently looking at is how the histones and chromatin organize themselves during this course of maturation. And with that, I'd like to thank my lab members and members of Dr. Clement's lab who've also been really helpful with this entire study, and of course the
Do you know how much they actually consumed and how much actual alcohol was consumed daily?
So we did do blood alcohol studies, and we observed that their mean blood alcohols were roughly in the range of 150 to 200 milligrams per deciliter. And in humans, anything above 80 is legally not allowed or to drive and operate instruments, but above 100 is binge drinking. So these mice were drinking regularly 150 to 200, and that also correlates well with other studies where they use the gavage model, and they see the same spike in alcohol. Yeah, pretty much.
A few years ago, Dr. Tim Cutt and Dr. Shannon and I did experiment with alcohol-induced fetal growth retardation in a sheep model. We gave this alcohol to the mother during pregnancy, and the fetal growth is all reduced, the brain development all impaired. And interestingly, we gave this the mother glutamine to prevent S-base imbalance, and everything improved. The fetal growth improved, the brain, the problem all disappeared. So I wonder if glutamine worked also in your male model.
That's quite interesting. So, in the maternal side of things, there's definitely more studies with alcohol, and there's correlations with Wnt beta-catenin signaling. However, in the paternal side, there's fewer studies, so there's not really consensus on where the changes are happening. But there are a lot of reports saying that interventions, like you were saying with glutamine, in this case, interventions, maybe lifestyle changes or exercise even, can reverse some of the effects, not in the alcohol especially, but in other exposures.
All right, we're going to go ahead and move on to the next speaker. Okay, our next speaker is Robin Moses from Dr. Bazer's lab, and Robin's the first one to send me an introduction, so I'll say a little bit more about her. She's from San Antonio. She got her BS in biology from Texas A&M and worked as an undergrad with Dr. Bazer, and then is now in the second year of her master's student. Today she's going to talk about the utilization of glucose and fructose by the ovine conceptus during pre-implantation period in pregnancy. Thanks, Robin.
Thank you, Dr. Fuller. Thank you all for being here today, and I'm quite honored to be able to present my research today. So thanks to years of extensive research, we now know that the early embryonic development period is considered to be a highly proliferative process, often with a high nutrient demand. For species such as cattle, pigs, and sheep, this proliferation is very pronounced before implantation can occur. For sheep specifically, the fertilized embryo will enter the uterus around 3 days post-fertilization and will then begin to elongate from a will then begin to elongate from a spherical structure to a tubular structure, which will take place around days 10 to 12 of pregnancy. During this time, the embryo will begin to develop the extraembryonic membranes, and from here on after, I will refer to this as the conceptus. The conceptus will continue to elongate from this tubular structure into a longer filamentous structure, which typically will extend from both uterine horns to increase surface area for apposition and adhesion, which will begin to take place around day 16 of pregnancy. And this is where our story is going to begin. Now, due to the increased nutrient demand, a lot of research has been focused on the role of glucose during this period, which makes sense because glucose is one of the most utilized nutrients in the body. Through glucose metabolism via glycolysis, we can get two molecules of pyruvate, which can further be metabolized. If oxygen is available and if the nutrient demands require it, it can be oxidized through the TCA cycle to produce electron carriers such as NADH and FADH2 for oxidative phosphorylation and ATP production. If oxygen is not available, it can be reduced down to lactate via anaerobic metabolism to regenerate NAD+ for glycolysis and recycle lactate back to pyruvate. Now, glycolysis itself can be an entry point for other pathways. For example, the first step, phosphorylating glucose to G6P, can be an entry point into the pentose cycle for the production of ribose and NADPH. And later on down the pathway, 3-phosphoglycerate can be utilized for serine and glycine biosynthesis. Both of these pathways are very important for the production of purines and thymidine for DNA synthesis, which is needed to keep up with the rapid cellular division. Glucose can also be metabolized to fructose via the polyol pathway, and fructose can enter glycolysis directly. Glucose and fructose are also important substrates for the hexosamine pathway, which will be important later on during pregnancy. Here it is. Now, as I had mentioned, both of these substrates are very important throughout gestation, and as we can see from these graphs, both glucose and fructose increase in the allantoic fluid with gestational days. The allantoic fluid is considered to be a nutrient reservoir instead of just what we would consider fetal waste at this point. What's interesting to look at is later on in gestation, we do see that fructose is 11 to 30 times greater than glucose, which leads us to wonder if this could be a possible metabolic substrate for the fetus development. So our question became, can the ovine conceptus trophectoderm use both glucose and fructose before implantation can occur to support the conceptus development? And then can these products, the metabolic products, be used as substrates for other pathways? So for our experimental design, we synchronized our ewes using a progesterone seeder and left it in for 2 weeks, removed it, administered Lutalize, and then bred them to fertile rams at day 0. 17 days later, the ewes were euthanized and the uterine horns were flushed with phosphate buffered saline to collect the conceptus tissue. We also collected endometrial tissue and a blood sample for further analysis. That same day of necropsy, we performed an isotopic tracer experiment. To do this, we cultured, oh no, that's not supposed to happen. Okay, we cultured about 20 milligrams of the conceptus tissue in a isotopic mixture with a radioactive glucose or fructose and then unlabeled substrates. We then gassed it with 95% oxygen and 5% carbon dioxide, incubated that at 2 hours at 37 degrees Celsius, terminated the reaction with perchloric acid, and then measured degradations per minute of any detectable carbon dioxide produced the following day. And just to orient you to the experimental setup, what we did was we had the conceptus tissue in the reaction mixture with a center well bucket suspended in the tube with a small PCR tube sitting inside of it. And the hope was, as metabolism occurred, the carbon dioxide would kind of float up into this tube and be captured. So we would then remove that tube and place it in a scintillation cocktail to be, for the degradations per minute to be measured by a liquid scintillation counter. And the treatments that we used was a universally labeled glucose or fructose along with a unlabeled substrate. For our first treatment, we had equimolar glucose and fructose, and we also had a condition which was fructose only. We then had a similar treatment for the universally labeled fructose and a fructose only treatment as well. What we ended up seeing, as expected, was an increase in detectable carbon dioxide produced from our glucose-only treatments. This makes sense because there's no fructose to compete with it, so we would see the highest production from this group. Our next highest production was the universally labeled glucose, as well as the unlabeled glucose and fructose. This one's a little bit more stunted, which could be attributed to the fact that fructose is there and could possibly be a substrate, but we wouldn't be able to detect it because it's not labeled. Our lowest group is the labeled fructose and the fructose and glucose treatments. Here we are seeing a smaller bar because glucose is there as a competitor. Just to kind of explain this, fructose can be metabolized by hexokinase to produce F6P, which is a substrate for glycolysis, but in the presence of glucose, this G6P molecule produced by glucose and hexokinase will block any entry of fructose into this pathway, thus smaller bar. The treatment of interest is this fructose-only group. There is no glucose available for the conceptus to use, but the fact that we are seeing some detectable carbon dioxide being produced suggests the conceptus can utilize fructose in the absence of glucose. We then wanted to see if this day follows a similar pattern as what we would see later on in pregnancy of a increased fructose production. So in day 17 uterine flush, what we do see is an increase in fructose production of about 15 times higher than glucose. We also wanted to see what pyruvate was doing at this point, so we performed an assay using lactate dehydrogenase. And lactate dehydrogenase can interconvert lactate and pyruvate based on the cellular needs or oxygen availability. So if oxygen is not present, it can be reduced down to lactate, versus if oxygen is present, it could be converted back to pyruvate. And what we saw was no significant difference between production of lactate or pyruvate across any of the treatments. However, I would like to point out the scale bars over here. We are seeing about 15 nanomoles of lactate produced per milligram versus about 90 nanomoles of pyruvate produced per milligram. This is about 15 times higher production of lactate to pyruvate. We don't know for certain if this is because all that lactate is being produced due to the uterus being a hypoxic environment, or if that pyruvate is being utilized right away into a different pathway. It would require more investigation. So in summary, the ovine conceptus can metabolize both glucose and fructose, but does prefer glucose over fructose. And the conceptus tissue can also convert glucose and fructose to pyruvate and then subsequently lactate. However, further investigation is needed to determine contribution of these two substrates into different metabolic pathways. And to do this, we are currently in the process of breeding our next round of sheep for a specifically labeled isotopic tracer. We will be using a carbon 1, which can be utilized. The carbon dioxide will be pulled off in the TCA cycle and the pentose cycle. And we will also be utilizing a carbon 6, which is the carbon dioxide will be pulled off in the TCA cycle only. So then to determine pentose cycle contribution, we would subtract the carbon dioxide from the carbon 6 from the carbon dioxide from the carbon 1, and we would be able to see what's going through the pentose cycle. We also are planning to quantify messenger RNA expression in the conceptus and endometrial tissues for key metabolic enzymes, as well as for IHC and IF on these same candidate genes, proteins. And with that, I would like to thank Drs. Baser, Wu, and Johnson for their guidance and knowledge for this project, as well as Drs. Hewan Stow and Claire Stenhouse and Avery Kramer and Kitty Halloran for all the hard work on their help on the project. And I will take any questions at this time.
Questions?
Is it that the fetus prefers glucose, or the mother can make glucose earlier? Like, have you looked or considered that potential angle? Like, maybe just early in pregnancy, the mother has more capacity to throw that molecule out.
So when the mother transfers the glucose over to the fetus, it can convert it to fructose, and once that fructose is sequestered in the allantoic fluid, it can't be transported back to maternal circulation.
Okay. Okay.
Other questions? All right, we will thank Robin. And then Marcus.
What's the best way to do this?
Go ahead and do that here.
After I'm done. Okay.
Okay, so we'll move on to our next speaker, who is Marcus Arzabal from Dr. Jan Thrombidos's lab in the vet school. Make sure this loads.
I guess not.
All right, so howdy. My name is Marcus Arzabal. I'm a third-year PhD student in Dr. Ramadas's lab. We are a perinatal research group that focuses on the physiological effects of different teratogens and how that affects the development and pregnancy for both the mother and the fetus. Today I'll be speaking about our studies on the impacts of e-cigarette aerosols exposure during pregnancy and how that affects development. So, nicotine is a highly addictive substance that's found in tobacco products. Tobacco product use during pregnancy has been known for years to have detrimental effects on the fetus. However, about 50% of global daily smokers will continue to smoke during pregnancy. So now with the invention of electronic nicotine delivery systems, it's just another way of obtaining that nicotine. And so you can see from this figure down here that there are several different forms but they all pretty much do the same thing. And they also come in different generations. So as time has progressed, the ability for these devices to deliver nicotine has actually increased, so they can deliver higher rates of nicotine as well. There's also a perception that these devices are safer to use since they do contain less chemicals than traditional cigarettes. However, safer is not always safe is the wording that my little sister told me actually from her high school teachers are now telling students, safer does not mean safe. So I kind of like that. But largely, there is not enough studies done currently to prove that these are safer or that they are safe. So vaping is essentially you have a e-cigarette liquid that contains propylene glycol, glycerol, nicotine, and flavorings. And what happens is they are heated to a really high temperature by a metal foil, and the suction created by the user draws the liquid through a cotton filter, and that aerosolizes the liquid. And then you see that this aerosol is produced and kind of mimics smoking. So that rapid heating and vaporization of the liquid actually releases a larger number of chemical constituents than what is originally found in the vaping liquid. Right now, all the chemicals that are listed up here are approved by the FDA for oral ingestion. However, they are not approved for inhalation, which is kind of why regulations have been sort of tricky. And it's only now that they are starting to be either banned or regulated by local, state governments and some businesses. So there are no current longitudinal studies just due to the nascence of this research field. So this research is really important because a lot of people are using e-cigarettes across the world. The few human observations that they've seen for e-cigarette aerosol exposure in pregnancy has shown reduced extent of damage compared to traditional cigarettes. However, they are still seeing effects. And this figure here is from a review article that we published basically describing all of the recent findings on e-cigarette aerosol exposure and pregnancy. They've only been able to use animal models to identify neurobiological, pulmonary, and cardiovascular outcomes. And so we have contributed to both pulmonary and cardiovascular at this time. And the delivery method is going to be really important for these animal models because we want to basically mimic how a mother would be exposed to the e-cig aerosols as well. Now, a lot of these models have been using intraperitoneal injection or an oral gavage to expose the mother and the fetus to these chemicals. So what our model is a environmental whole body exposure. We use this e-cigarette chamber that's custom engineered. So we have several of these chambers, and we're able to expose all of our groups at the same time. And for the same, we can expose them to the same concentration and same amount of vapor as well. We have this software interface that essentially fires the atomizer to generate e-cig aerosols that flows through the tubing into the chamber and then out through a filter into the facility exhaust ducts. When we sampled aerosols from our chambers, we found that there was propylene glycol, glycerol, nicotine, but also a large number of other chemicals that weren't present in the liquid to begin with. We excluded flavorings from our studies because the flavorings in commercial e-cigarettes are extremely variable and in concentration and flavor. They come in menthol, tobacco, bubblegum, cherry, anything, you name it. They probably have a flavor for it. So we decided to exclude that so we can focus on the effects of the main components of the e-cig liquid and how that affects pregnancy development. Our model is a binge e-cig exposure model. So we use a 3-hour a day, 5 days a week exposure time, and that's from gestation days 5 through 20. And this produces blood nicotine levels in the mothers that are similar to moderate to heavy smokers. So, in order to test the effects of these e-cig aerosols on growth and development, we used 3 groups. We have a juice plus nicotine group, a pair-fed juice group, and a pair-fed control group. The juice is just referencing the propylene glycol and the glycerol. So a term commonly used by e-cig users is a e-cig juice, is referring to the liquid. And so we have an e-cig liquid with nicotine, a pair-fed control group that uses just the e-cig liquid with no nicotine, and then a pair-fed control group that is receiving only room air for the same period that the 2 treatment groups are receiving their aerosol treatment. So, with our nutritional control, we found that the mothers had a consistent maternal weight at the 2 time points that we checked for fetal growth and postnatal growth. We found that in the gestational day 20, there was no difference between the control and the pair-fed juice group. However, there was a significant difference between the juice plus nicotine group compared to the control and the pair-fed juice. And that is carries over also to the crown rump length. So we have, again, no difference between the pair-fed juice and the pair-fed control, but we do have a significant difference in crown rump length between the juice plus nicotine compared to the controls. So we also measured growth parameters at postnatal day 10 and found that there was a significant decrease again in both the weight and the crown rump length compared to controls and the pair-fed juice controls. And then during the postnatal study, we measured weights daily from postnatal days 1 to 10 and found that the first 2 days or 3 days, there was no difference, although they were a little bit lower between the e-cig juice plus nicotine groups and the 2 control groups. However, after that, their growth was significantly reduced throughout that postnatal study. So, because growth restriction is commonly associated with reduced blood flow to the uteroplacental compartment, we wanted to look at some of the hemodynamics of the mother and the fetus. So we looked at maternal heart rate and fetal heart rate and found that there was no difference between the 2 or between any of the groups. And then we looked at blood flow through the maternal uterine artery and the fetal umbilical artery. And we found that there was a significant reduction in the juice plus nicotine maternal uterine artery blood flow. So there's reduced blood flow to the uterus, and then there's also reduced blood flow through the umbilical artery within the fetal compartment. It is interesting to note that while not significant, there is a slight reduction in the pair-fed juice group, so it's kind of a descending pattern that we're seeing. So because we've noted there is significant growth deficits associated with e-cig aerosol exposure during pregnancy, we also think that there is going to be significant detrimental effects to the development of other organ systems. So this could include the lungs, the brain, the muscles, skeleton, everything. So what we think is that there could be increased risk for sudden infant death syndrome and infant respiratory distress, neurodevelopmental effects, increased risk for adult onset diseases, and an increased risk for epigenetic changes, which are also noted in traditional cigarette smoking. So kind of keeping with the initiatives of the Heart, Lung, and Blood Institute, we want to identify molecular pathways within the reproductive vasculature to identify why we're seeing these reproductive vasculature deficits. And we also want to look at next exploring the alterations that could be possible to the pulmonary and cardiovascular system of the offspring. And so what we're doing now is we're looking at the lungs of these pups at postnatal day, gestational day 21, one day before they're due to be born. And we're looking at different morphological effects right now. And we are finding that there is some differences between the control and both the e-cig juice group and the e-cig juice plus nicotine group. So there is some effect that is noted whenever we have the e-cig aerosol exposure without nicotine. And so that's going to be really important because we need to identify what mechanisms are caused by the e-cig aerosol as a whole and what is already caused by nicotine. And so I'll be really excited. Hopefully I'll get to share this with you maybe at the next IFRB whenever we do finish these studies. It'll be really interesting. I'm really excited to work on this. But until then, I just wanted to acknowledge my lab, my mentor, Dr. Ramadas, my co-workers, Matt Nemick, Vishal Nayak, Katie Davis-Anderson, Rain Young, Lundy Young, Josue Ramirez, Jaehoon Lee, our collaborators at Texas A&M, our external collaborators, and our funding sources, NIH and IAAA, Texas A&M, Research Society on Alcoholism, and then, of course, the IFRB committee for allowing me to come and speak today. So, any questions?
Questions?
So I don't know if I caught it. Maybe you said it. What was the percentage nicotine that you were giving them?
So we're using a percent that's 5% and 10%.
Okay.
So we have
Just like the pods that they would give?
Yes, exactly.
Okay, cool.
Other questions?
That is all pulled. So we aren't seeing any sexual dimorphisms between the two. They're both reduced.
Any other questions?
Right. Mm-hmm. So right now, so that's a great question, because right now we are only following postnatal 10, but it would be really interesting to see how long those growth deficits last. I'm not sure, but it's something that we plan to look into.
All right, let's thank Marcus. All right, well, I pull up our next speaker here. Just make sure just that you've signed in the thing in the back as we move toward lunch. Our last speaker before lunch is Tatiana Maya, and Tatiana did a undergrad degree in Brazil, and then came to the U.S., did an internship with me when I was in Tennessee, and now is a current master's student with Dr. Williams and Dr. Cardosa. And today she's going to talk about does prenatal and postnatal nutrition interact to program reproductive phenotype in sexually mature heifers. Thanks, Tatiana.
Okay, thanks for the introduction. So today I'm going to be presenting some preliminary data from my master's project, and we want to see if pre and postnatal nutrition interact to program reproductive phenotype in sexually mature heifers. So the lifetime productivity of beef cows is influenced by their ability to reach reproductive maturity, to conceive, to sustain pregnancy, and to calve by around 24 months of age. And one of the factors influencing those characteristics is the nutritional management. A study published by Berker in 1993 showed Becker and colleagues show that gestational and early infancy starvation in humans are associated with increased risks for cardiovascular and metabolic disease in adulthood. So this study, in conjunction with others, demonstrate that the perinatal period, which is the period immediately before birth and after birth, is a window of susceptibility for the negative impacts of an adverse environment. And also, nutritional perturbations during this period can cause permanent and adverse effects on the organization of neuronal pathways. And our laboratory has shown that nutritional status during the juvenile period, which is between the 4 and 8 months of age, affects important neuroendocrine signaling pathway that regulate GnRH secretion, also affects DNA methylation and timing of puberty. So these findings from our laboratory and another studies, we believe that organizational changes brought to the pre and postnatal nutritional programming is imprinted in the genome, and it's most likely to be associated with consistent physiological events that can be manifested only later in life. So therefore, a better understanding of this process can allow us to develop strategies to optimize lifetime reproductive performance and animal health. So currently we have a project funded by USDA, and it's a long-term project, and we are analyzing several of other characteristics, and we want to study the impact of pre and postnatal nutrition on reproductive neuroendocrine phenotype in sexually mature heifers. So the overall hypothesis of this project is that the maternal nutritional environment during the mid to late gestation and associate with either elevated or restricted energy intake postnatally, it will interact to direct long-lasting changes in adult reproductive phenotype. So in order to get that project started, we had a total of 108 Brangus and Brayford beef cows pregnant with a female fetus, and they were previously inseminated with a single semen of Hereford or Angus sire, and then the ones that were not that did not conceive by AI, they were bred with a cleanup sires. And so by 3 months of gestation, those cows were fed to achieve a target body conditioning score by 6 months of gestation and maintain until calving. So here we can, So here we can see that they're stratified in those three groups, and the low group, they were fed to achieve a between 3 and 3.5 body conditioning score, and then the moderate 5.5 to 6, and the high 7.5 to 8. So those are representative cows after they had achieved the body conditioning score that we want. And here you can see that as soon as they reach the body conditioning score that we want, they were just fed to maintain that body conditioning score, and the same happened with the weight. So once those heifers calves born from those cows, they're weaned at 3 to 3.5 months of age, and after being acclimated to a diet for 2 weeks, they're stratified by age and body weight, and then assigned randomly within maternal group in 2 dietary treatments until 8 months of age. So they're assigned to a low gain group where they're fed to gain 0.5 kilogram a day, and then a high gain group where they're fed to gain 1 kilogram per day. And once they have reached 8 months of age, those animals are placed on common growth diets until puberty. And then by the time of 8 months of age, those heifers, when we compare the high gain group with the low gain group, we saw that the high gain group were on average 100 kilograms heavier than the low gain group. So by the end of this treatment, we would have a combination of the maternal treatment with the postnatal treatment, giving us a 3/2 factorial, and we would have a total of 6 treatments and representing 18 heifers per treatment. So we also, during the period that both cows and then the heifers were having this nutritional programming, we measured the plasma leptin, the plasma concentrations of leptin, and then we can see here for the maternal group, the leptin for the obese group start to increase, and then once they have reached their body conditioning score, we start decreasing their diet. So the leptin concentration started to decrease. And the low and the moderate, they had similar concentrations. And then for the high for the heifer, the high gain group had a higher leptin concentration compared with the low group. And also, when we analyzed a data from about 2/3 of those heifers, we saw that there was effect on postnatal treatment and maternal and postnatal treatment interaction on age at puberty. So when we look at the postnatal treatment, the high gain heifers, they reach puberty 62 days earlier than the low gain heifers. And when we look at the interaction of treatments, the high heifers, they reach puberty 92 days earlier than the low. So my master's project, our hypothesis is that the nature of postnatal diets during the juvenile period will either exacerbate or mitigate the negative effects of prenatal nutritional extremes. And the objectives of this study is to determine the effects of pre- and postnatal nutrition on phenotypic characteristics of the estrus cycles of sexually mature heifers. So we look at expression of estrus, ovarian follicular reserve, ovulatory follicle size, and peak of estradiol secretion, corpus luteum function, secretion of progesterone, estrus cycle length, and endometrial thickness during the periovulatory period. So the experimental design had a total of 42 sexually mature heifers, and they were representing all the 6 combinations of groups that I just described. And we had a total of 7 heifers per treatment, and they were assigned in 4 groups, having 10 to 12 heifers per group. And currently, the data that I have collected in the analysis regards to 32 out of those 42 heifers, and this is what I'm going to be presenting today. So we start synchronizing those heifers, giving them 2 injections of prostaglandin 11 days apart. And on the day of the second prostaglandin injection, I start ultrasound those animals, and I ultrasound first until ovulation for the cycle that I call PGF cycle, and then for the following cycle, I ultrasound then starting from day 14 until the day of ovulation. And we, in addition to the ultrasound measurements, we also daily blood samples those animals, starting from the second injection of prostaglandin and going from PGF cycle 1, PGF cycle 1, and cycle 2. And then the cycle 3, we just use to look at the day of ovulation and estrus behavior. So here is some images that I have from the ultrasound, and so we measure the dominant follicle growth, also the corpus luteum size, and the relative endometrial thickness, ovar size, and then here it's a video of the follicle count. And so to increase our accuracy, I measure those, I count those follicles per heifer in 2 cycles, and then you can see, so I count each of those follicles that were bigger or equal to 3 millimeters. And for the blood samples, we analyze the progesterone and estradiol concentrations, and then for progesterone, we use a progesterone kit with standards formulated in fetal bovine serum, and for the estradiol, I'm still working on this data. So all statistical analysis was performed using JUMP, And we used two-way analysis of variance for factorial design with interaction, and also mixed model analysis for repeated measures. So according to those preliminary results, we didn't see we didn't see any significant main effects or interaction on the variables listed in this table here. So you can see that the p-values were all high, and those are just the means from each of those variables. So when we look at the progesterone concentration using mixed model, we saw there was a significant effect of treatment and the interaction with p-value less than 0.05. So we also decided to analyze the progesterone concentration using the area under the curve. So I analyzed starting from day 0, which was the day of ovulation, until day 14, which was the day that we still believe that the CL was function on all those animals. So based on that data, we saw there was a significant maternal effect and also a treatment interaction effect with a p-value less than 0.01. And when we look at the maternal effect, the low group had the highest area under the curve, and it was different than the moderate group. And when we look at the treatment interaction, we saw that the low-high heifer had the highest area under the curve, and it was different within the other treatments, and then the other treatment was similar. So that's, you can see all the letter B here represents that they're similar. So we want to demonstrate what's going on, and then since we saw there was a maternal interaction here, and this group that represents the highest, that when you compare those two, you can see that there is a more contrast on the side. So we decide to demonstrate by creating a plot with a line graph here, so to see where the difference is coming from. And then when we compare the low-high heifer with the moderate-high heifer, we saw that they're actually the concentration, the progesterone concentration of the low-high heifer is greater. It's greater than the moderate-high heifer. So we see that even those heifers, they had high diet postnatally, because they had a maternal treatment effect. We can see that results here, represented by this graph. So again, these data are preliminary, and we still are working on the results. And I come back with the question of of the title: do pre and postnatal nutrition interact to program reproductive phenotype in sexually mature heifers? So we can't forget that based on what I present here, when we analyze the age at puberty, for those heifers, we saw there was an effect, a treatment effect. But once I studied the estrous cycle of those mature heifers, we couldn't, it doesn't seem that there is a major effect on those characteristics that I evaluate, even though we can see progesterone difference when you look at the area under the curve. So I will complete my data next year, and hopefully can come back here, give more accurate results for you. So with that, I would like to thank you, USDA, that has been funding our project, and also everybody from our lab, and everybody involved also, because I was in Beeville. So every people here, they're involved in this project. And I want to thank you guys. And with that, is there any questions?
All right, one question in the back. Just go ahead and.
You mean for the maternal, or sorry, the heifers? So yeah, so we look at the body condition score just when they're pregnant. But so we starting from the 3 months of gestation. This is when we actually assign them for those to feed to achieve those body condition score until by the end of pregnancy until Kevin. But then I don't have the information after this for the body condition score of those months. Yes, so there is a attendance for the low group, you know, by they're gonna have a really a high gain in order to compensate, you know, but you can see that usually the low group they're less heavy than the high group. They're smaller also.
Hey, Tatiana, I have a question. So in that you're dealing with a subspecies cross here of Brangus and Brayford, and we know that the subspecies in Tyrus indicus have differences in follicular waves and
Yeah.
follicular function and even CL development, progesterone clearance, etc. Being that you didn't see any differences in any of those, do you think your cross is consistent enough that you have an equal amount of indicus and Tyrus and that you sort of wash all that out, or do you have any comments about that?
Well, I think, I mean, the reason that I see, we expect to see some differences, but the reason why we still haven't seen a lot of differences also because I don't have all enough animals, but I think that's not. I think it was the same amount, and this is not something that would intervene in this experiment. Okay.
Class, make a few comments.